Hello, my name is Izabella Zamora. I'm currently a master's student at MIT and the Broad, and today I'm going to be talking about how we've approached understanding spatial organization of cellular plasticity in PDAC. These are my disclosures. So first off, what is plasticity? We define plasticity as the ability for a cell to transition between states. For example, the epithelial to mesenchymal transition, which leads to things like invasion, metastasis, and therapy resistance, which all correspond to poor patient outcomes. So we would like to understand not only these states' profiles, but also what are the transition dynamics between them and how do extrinsic factors influence these transitions. To best address this question, we are going to use spatial transcriptomics. We used COSMICS, which is a single molecular imaging technique that captures transcripts at a single cell resolution. One of the key challenges of this type of data is the limited gene panel. In the data we will be discussing today, we only have 960 genes, which is insufficient to use the same clustering approaches that are used in single cell data to resolve fine states, such as our plasticity substates. So to overcome this challenge, we leverage both spatial and transcriptional information for each cell. We hypothesize that cells near each other are more likely to be clonally related and that neighbors of cells tells you something about their own state. So just to reiterate, our goal here is to understand extrinsic factors influencing plasticity using spatial transcriptomics data. To do this, we developed PlastiNet. PlastiNet takes in spatial coordinates, expression, and inferred ligand receptor analysis results with the potential addition for other multimodal inputs. This feeds into a graphical attention network, which is a deep learning-based method similar to neural nets, except we have the addition of attention. Attention has been proven over the past couple of years to be a very powerful tool in deep learning, enabling tools such as ChatGPT to weight how important different elements of an input are and better understand global relationships. This graphical attention network acts to reduce the inputs from each cell into an embedding that encapsulates spatial and transcriptional information for each cell. We can then cluster this spatially aware embedding to define neighborhoods and also identify plasticity states, as well as infer their transition rates. To benchmark PlastiNet, we compared against other existing neighborhood analysis methods, and we used slide tag brain data because of the known structure and characterization of the brain. PlastiNet is able to perform just as well, if not slightly better, than other state-of-the-art methods, as shown by our benchmarking results, showing good spatial conservation as well as high niche coherence scores. Next, we focus on PlastiNet's ability to reconstruct differentiation paths in the colon. We use the colon as a model organism in this case because of its well-defined differentiation path from stem cells in the crypt all the way up to differentiated cells in the villi. Not only do you see a transcriptional gradient from stem cells to differentiated cells, but you also see a spatial gradient from bottom to top. PlastiNet was able to reconstruct the expression gradients over this differentiation path. The bottom of this heat map shows early pseudo time points, high in stem cell markers, the middle, high in this transitionary marker CD24, and the top, high in differentiated markers. We then plot the predicted differentiation path on our spatial data, and we see that there is a clear spatial gradient from the crypt up to the villa, and that PlastiNet is able to reconstruct a differentiation path that makes sense transcriptionally and spatially. So now we move on to apply this to our PDAC data. First, looking at neighborhoods that were identified, we can see that there are very distinct neighborhoods in this tissue sample. Here, I'm only showing one piece of one of our slides, but the analysis was done across 3 full slide samples as well as many TMA sections. Taking a closer look at some of these neighborhoods, these are our immune neighborhoods with the dot plot showing their composition. And we see that there is a global immune neighborhood with lots of different immune subtypes, a CAF-enriched immune neighborhood, as well as this small macrophage and T-cell enriched neighborhood. And we hope in the future to subcluster this further to get smaller immune neighborhoods, such as tertiary lymph node structures and potential immunity hubs. I would also like to point out these 2 cancer neighborhoods that we found. There is one cancer neighborhood shown in purple that is majority cancer cells, and then there is a more immunosuppressive cancer neighborhood shown in green that is high in macrophages and SPP1, as well as cancer-associated fibroblasts. We then construct a differentiation path in the tumor cells from classical to basal. We see in early time points of this pseudotime, you have high expression of classical markers that decreases over the pseudotime, where you see the opposite for the basal genes. And then we discretize along the pseudotime into 5 distinct plasticity states, ranging from classical to basal, with 3 intermediate states in between. And going back to the neighborhood results, I would like to mention that the classical states were found in the tumor neighborhoods, whereas the basal states were found primarily in the immunosuppressive neighborhoods. Now that we have defined plasticity states, we want to better understand their transition dynamics, which we measure using spatial transition rates. We're operating under the assumption that cells that are close in space are more likely to be clonally related than cells that are distally apart. And so we use random walks on the spatial tissue graph to estimate transition rates. We see that classical cells have the highest probability of staying in the same state spatially, suggesting spatial autocorrelation, and that along this classical to basal plasticity, we see that cells pass through the intermediate state with very little skipping. Now that we have these states, we want to understand the extrinsic factors influencing them. So to do this, we first looked at what cell types were enriched or depleted over the null distribution in each of these subtypes, and we saw that classical cells were enriched in plasma, B, and monocytes, whereas intermediate cells were enriched in CD8 T cells, NK cells, and ductal cells. And finally, the basal cells were enriched in more immunosuppressive cells, which we expect based on literature, such as cancer-associated fibroblasts, SPP1 macrophages, neutrophils, and mast cells. Beyond co-localization of cell types, we wanted to understand how these cells interact with their tumor microenvironment. One potential mediator of spatial plasticity that we were able to identify is CD44 on tumor cells. We saw a steady increase in CD44 expression from the classical to intermediate to basal states. In the classical cells, we see that CD44 is receiving signal from all sorts of collagen in its environment. And then in the intermediate state, we see that ductal cells are sending SPP1 to the intermediate cells. And in the basal state, the basal cells are producing SPP1 as well as MIF, which stimulates macrophages to produce more SPP1. And so we hypothesize that there's potentially a basaloid population self-loop through the amplification of SPP1 signaling that either maintains its state or maybe progresses the intermediate state into more of a basal state. Overall, we have a lot more work to do to further explore and validate some of our findings, but here we show that PlastiNet, okay, sorry. PlastiNet is able to better understand spatial transcriptomics data as validated by reconstructing spatial neighborhoods in the brain and understanding cellular differentiation paths as validated in the colon. And when applied to PDAC, PlastiNet is able to identify several tumor and immune neighborhoods, as well as finally resolve a spectrum of classical to basal plasticity and estimate transition dynamics. We show that it can be used to generate hypotheses from patient samples and present CD44 as a potential mediator of basal to classical plasticity. Thank you so much for your attention. It's an honor to be here today. I would like to say thank you to Dr. Neerha Cohen and Arnav Mehta for their support throughout this project, as well as David Ting and Bidish Patel for the data, and members of the Cohen and Broad in general, all the funding sources, and most importantly, the patients and their family that made this work possible. Thank you.
Questions for Izabella, please.